Very good morning to all of you. So, in today's lecture, we are going to see about a different topic which also will be a part of your preparation for NEET, a very important one. So, this is pressure in liquids. I hope I am audible to all of you because I just saw one or two messages where they are saying there is no audio. So, I think there is some problem from your end, Madhu. So, it would be problem from your end, kindly get it fixed. Okay. So, we will see today about something called pressure, and as you have been seeing that in different classes, I have been telling a few things that generally before knowing in detail, we have some misconception about a few topics, so that should be cleared, right? So, before it is too late. So, similarly there exists a misconception about something called pressure. So, I have heard many, they keep on saying that pressure is exerted in this direction, that direction and so on. So, that thing has to be broken. So, pressure cannot be exerted in this or that direction, it is not a vector quantity, it is a scalar quantity, right. The force which is causing the pressure or creating the pressure is a vector, understood. So, here what we are trying to say is, first when we define pressure, then you should know what is the definition of pressure. Pressure is defined as thrust force. Whenever we say thrust force, what is thrust force? It is a push, it is a pushing force, right. So, thrust force per unit area of application. So, when we take thrust force per unit area of application, that is known as pressure. So, when we have this term, that is the thrust force, so as I told, what we should be clear with, that thrust force is a push, and this is always normal to a surface or cross section at every point. So, when we have a force that is perpendicular or normal, and second thing, it is a push force. So, that force per unit area of application is called pressure. Next thing that we should be clear with pressure is that pressure is a scalar and is an effect of force. So, do not be confused with the direction and all, okay. Do not be confused with pressure is exerted in that direction, it is a vector. No, it is not a vector, okay. So, scalar quantity and an effect of force. When force acts on a body and it tries to rotate about any axis, we define another effect which is called as torque, understood. In a chapter called elasticity, you will study when force tries to deform a body, then due to that force an effect is created inside the body which resists the deformation, it is called stress. Similarly, when a force is acting normal or perpendicular to a surface trying to push it, so the effect is known as pressure. Is it clear to all of you? So, when we talk about the average pressure exerted on an area A, so average pressure is the total thrust divided by area of cross section or area. So this is how we define pressure, and as I said, it is a scalar quantity. Clear to everyone? Okay. Now this pressure has got several units. When we talk of its units, in SI system you have force by area, which is Newton per meter square, or also known as Pascal, PA. So this is SI unit. Other units you have dyne per centimeter square, which is CGS unit. There are other units as well. You have ATM, that is atmospheres. Apart from that, you have bar, you have torr, named after the scientist Torricelli. You have centimeter or millimeter of mercury. You also have unit PSI, pound per square inch. Okay, so this unit is also quite common, PSI. So every pressure, not only fluid pressure, Farhin, listen carefully, not only fluid pressure, every pressure is thrust by area. Okay, and second thing what I am saying is there are different units, Newton per meter square or Pascal, both are SI unit. Dyne per centimeter square, which is the CGS unit, atmospheres, ATM, bar, torr, these are also different units. Centimeter or millimeter of mercury. Apart from that, PSI, pounds per square inch. Okay, so when tires are inflated in our vehicles, you will see that when you go to get the pressure over there, you will see they measure in PSI. So there are different units of pressure. This is clear to all of you. So you have to remember these are some important units of pressure, and out of this 1 ATM, this value is approximately 10 to the power 5 Pascal or 10 to the power 5 Newton per meter square. So 1 ATM atmosphere is also an important value, so that you should be remembering. Clear to all of you? Okay, so let's move on and kindly focus on the class. See, we as teachers, we are doing our job. Rest of you who are wasting your time by asking from where you are, from where you will be going, just think about where you would be going in your future. Do you want to get into a good college and get into a good profession? If you are aspiring to be a doctor, whether you want to be a doctor or you want to waste your time on social media or become a TikToker or YouTuber, whatever, that's your wish. According to that, you can pursue what all hobbies you are having presently. But if you are serious about pursuing your dream of becoming a doctor, so kindly focus on the class. Rest we can just tell, it's totally up to you. Okay, so we are talking about pressure, so I thought that this is the good time that we tell about. We don't want to pressurize you, even if we are teaching pressure, but we won't be pressurizing you. No one can pressurize anyone in this world. Okay, those who are destined to waste their time, they will do the same. Those who are destined to carve a niche for themselves, become good in their lives, become successful in their lives, they will silently do their own work, right? So there will be plenty of distractive elements, distractions going on in the world. So it's totally up to you whether you give up to that pressure that is being applied to you, right? Or you better focus on your goals and the things that are being taught. As I said, all that offline classes and online classes, this and that, all those waste things, these are of no use. These are useless things. When you will come over here, whenever the classes begin, so when these chapters will be taught, if you have paid slight also attention over here, so suddenly you can recall that yes, these all things that time whichever teacher has taken classes, he told this thing, so quickly it will click, and that time the chapter will become easier for you to understand. Okay. Otherwise in the class also you will be wasting your time in exchanging pleasantries with each other, that what is your name, how are you, where are you from, okay, all these things will go on. So you better decide what to do. So this was my small piece of advice which I gave, rest is up to you to follow. Now let's move. When we see here about pressure, so talking about pressure in fluids, so what is a fluid? Fluid is anything that can flow. So when we talk about fluids, is something that can flow. That can flow does not mean that it will always flow. We can keep it bounded and keep it static also. That means you can store water in a tank or in a glass, wherever. Or if you drop the water from the glass or make a hole in the tank, or through a tap you make the water flow. So you can make it flow also, you can restrict its motion also. Similarly, you can enclose a gas in a container, right, and you can make it flow also if you create a hole in the container, or through a valve you can allow the gas to escape outside. So fluids is that can flow. That means they are collectively, liquids and gases collectively are called as fluids. Yes, correct, Ramnath, it is correct. So that can flow. So now when we are talking of the pressure in fluids, so here we will be restricting our discussion on liquids. So when we talk about liquids, pressure in liquids. Now if we consider a particular type of liquid which we will call as incompressible. What do we mean by incompressible? Incompressible means density is same everywhere. The density is not changing, so such type of liquid is known as incompressible. So when we have an incompressible, then what we can say, first thing, and if we consider static liquid, so pressure at any point will be called as hydrostatic pressure. We will call that pressure as hydrostatic pressure. So pressure at any point will be known as hydrostatic pressure. What next we can say, at any point pressure will have one value. P at any point will be called hydrostatic pressure, and will have a single value at a given point. So at a particular instant, at a particular point, you will not have more than one values of pressure. There will be a single pressure at a given point. Okay. Next thing that you should be remembering, that when we are considering hydrostatic liquid, means it is in equilibrium. Hydrostatic liquid, and this liquid is in equilibrium. Which type of equilibrium? Static equilibrium. It is not moving. So in that case, what we can say, that every point the forces will be balanced. The liquid particles at every point will be in equilibrium, forces will be balanced. Yes, because yes, forces will be balanced. So in that case, sometimes you will see that any portion of the liquid, if it is exposed or open to the atmosphere, so that means that point's pressure will be same as the atmospheric pressure. So what we will say, pressure at any point of the liquid which is exposed or open to atmosphere. So pressure at that point will be same as the atmospheric pressure. Pressure at that point will be equal to atmospheric pressure. Understood? Similarly, if suppose any point which is in contact or exposed to vacuum, vacuum means free space, there is no particle, nothing. If it is exposed to vacuum, then the pressure at that point will be same as pressure of vacuum. What is the pressure of vacuum? Please reply fast. What will be the pressure of vacuum? What will you say about the pressure of vacuum? Yes, correct. So it will be 0. So any point which is exposed to atmosphere, its pressure will be P atm, and this P atm has a value which is 1 atm or 10 to the power 5 Pascal at sea level. When we consider sea level, the atmospheric pressure over there is taken as 10 to the power 5 Pascal or 1 atmosphere. And whenever a point is exposed to vacuum, its pressure is 0. Clear? Now, next important thing is I will be asking in terms of or in form of a question, because I will make things clear. Okay, concept should be known first. So, can anyone tell that pressure, can it be negative also, or pressure is always positive? I am waiting for your reply. Can pressure be negative? See, we talk about two things. One is what we call as absolute and one is called relative. Now suppose I am asking you that money that anyone can have, suppose I am talking about money in your pocket or in your purse, can it be negative? Then I will reply, can money or say coins or the rupee amount that you have, can it be negative? No, it can be either positive or 0. Okay, it can be positive or 0. 0, it can be 0. If you don't have anything in your pocket, you will say 0. But this is what we call as absolute. Secondly, if I say relative, suppose you are having 10 rupees and your friend is having 15 rupees, then relative to you, how much is your friend having? Can I say that your friend is having 5 rupees relative to you plus 5 rupees? And can I say relative to your friend you are having minus 5 rupees? Both are having positive amounts, but with respect to each other you are seeing one is having positive, other is having negative. Is it correct or not? Yes. So that means whenever we use the term relative to something, then it can be negative also. So here when I said pressure, I did not mention which pressure I am talking about. So pressure also can be absolute or relative to something, right? Whenever we measure the pressure, we generally compare it with a reference value which is called as the atmospheric pressure. So our pressure which we are considering may be more than atmospheric pressure, may be equal to atmospheric pressure, or may be less than atmospheric pressure, right or not? If you go in the depths of a ocean or sea, then you will see that pressure will keep on increasing, okay. Myself Charan, please wait for some time, let me explain, then you will come to know what is wrong and what is right, okay. So when you go below the sea level, you will see that pressure will keep on increasing. At the sea level, it will be equal to atmospheric pressure, but when you go down, it will keep on increasing, it will become more. But when you go up, you have heard that when we go to mountains and all, there is a drop in pressure. So the pressure will become lesser, right? So when we compare the pressure with atmospheric pressure, we get a relative pressure which we called as gauge pressure. And when we take the total pressure of any point, then we call that pressure as absolute pressure, which can be positive only or at max 0 in case of vacuum. But when we compare it with atmospheric pressure, we get what is known as gauge pressure, which is a relative pressure. So what we see next point which I am telling is, there is a pressure called gauge pressure. Gauge pressure is simply the pressure at a point minus atmospheric pressure. When we take the pressure at a point minus atmospheric pressure, we call it as gauge pressure. This is known as gauge pressure. This pressure at a point, when we take the total pressure, this is called as absolute pressure, and this is the atmospheric pressure. So absolute pressure, this can be positive or 0, which case it will be 0 if it is vacuum, otherwise it is always positive. Whereas gauge pressure can be positive or negative or 0. So this thing should be kept in mind. Understood? For example, if I say at a point, if number 1, pressure is equals to 2 atmospheres, this means gauge pressure will be 2 atm minus 1 atm. Atmospheric pressure is taken as 1 atm, so it will become 1 atm. This is positive. If I take pressure at any point as 1 atm, then in this case gauge pressure will become 0. If I select another point and I suppose take if suppose pressure at a point is 0.2 atmosphere, so at that point the gauge pressure will be 0.2 minus 1 minus 0.80 atm, which is less than 0 or not. So when we compare, Sadhika, why should we take atm pressure as 1? Everything has been given a value or reference. We call it as reference. Its value is actually 10 to the power 5 Pascal. But when you take something, suppose you are measuring length by a scale, from where do you start taking the length? As 0 or not? That mark you give as 0. When you take anything, you have a reference. Like you are taking pH values, you take a reference 7, which you take as neutral. Above that or below that, you come to know whether it is basic or acidic. So everything has got a reference. So atmospheric pressure 10 to the power 5 has been given a reference value of 1. You cannot take it as 0 because it is actually acting. It's not 0. So that value is taken as a reference, which is taken as 1. Above it gauge pressure will be positive, below it gauge pressure will be negative. Understood this? And if any pressure at any point is equal to the atmospheric pressure, we will say the gauge pressure will be simply 0. Understood? Yes, gauge pressure is Tikshana, yes, gauge pressure is something like relative pressure. That's what I was trying to say. When you write pressure at a point, that is the absolute pressure, but when they ask you gauge pressure, then you are taking it relative to the atmosphere. Means atmosphere is also exerting pressure on Earth. So with respect to that, how much more or how much less pressure any particular point in a liquid is exerting, that thing will be known as gauge. Clear? So shall we proceed now? Okay. So this pressure, when we take pressure inside a liquid and that to hydrostatic condition, liquid is in static equilibrium, so the pressure keeps on varying. How does it vary? Variation of hydrostatic pressure with depth in an incompressible. Now onwards, remember, whenever you see the word incompressible liquid, means density constant. So how does the hydrostatic pressure vary? So now when this liquid is concerned, this liquid has a density, constant density everywhere, let's call it as rho. Now here, suppose I select 2 points, let's call the point as 1 and let's call this point as 2. These 2 points are having a difference of h between them. So 2 is deeper than 1 by h, okay, or 1 is shallower, so it is going up. Hydrostatic, see the word, Yashu, what is hydro? Hydro means here pertaining to liquid, and static means, you know, it's not in motion. So hydrostatic pressure is when the liquid is static and in equilibrium, then we will say that the pressure exerted will be hydrostatic pressure. No, all liquids are not incompressible, but whenever we study something, we study first the ideal cases. So we are seeing the ideal case and seeing the result that we get by that. Why do we consider everything ideal? We first read about ideal cases, then only we come to know that practical life is different from ideal situation, and in what way it is different we come to know. So whatever observations we are making, that will undergo some changes, right? So always we take ideal cases, you take smooth surfaces, you take body falling in under gravity, you assume G is constant, you consider there is no air resistance, but in practical life everything has got resistance, friction and all, but we consider first the ideal cases. Understood? So hydrostatic, ideal liquid, static liquid at equilibrium. Clear? So here we are considering 2 points 1 and 2, which is inside the liquid. Now this no, Gangu Raju, will this class be repeated after we come to campus? No, it won't be repeated. This is the last time you are being taught, and directly after this, tomorrow they will be arranging special NEET exam for you. Okay, after giving that, you can directly become doctors. So nothing will be taught again. So these are some questions keeping on appearing every now and then. So a direct answer you can circulate it to your friends also. So everything is last. After this, directly you will be asked to sit in the NEET exam. Okay, fine. So rest of you can pay attention over here. So when this is in equilibrium, what we consider? We consider an imaginary cylinder of liquid contained between the points 1 and 2. Since this liquid is in equilibrium, we are considering a cylinder of liquid, because we have to find the area on which the force is acting and all. So what we do, considering an imaginary, we are assuming a cylinder type of structure, imaginary cylinder of liquid contained in between the points 1 and 2. You can consider any shape of liquid, we are considering cylinder over here. So just see. So we are considering an imaginary cylinder of liquid which I am drawing like this. Now what will happen? This is the point 1 and this is the point 2. So we have this liquid cylinder. This is totally a liquid cylinder in between these 2 points, and you can see that the height of the cylinder is h. Now since every portion of the liquid is in equilibrium, so this cylinder will also be in equilibrium. What all forces will act on it? Now if you see from bottom, suppose here pressure at point 2, let's take the pressure at point 2 as P2. Here the pressure is P2 and here the pressure is P1. So I said pressure is an effect, not a force. Now the force which will be acting by the liquid below this, it will push it up. Pressure force is always pushing force. So the thrust force due to P2 will push it up, thrust force due to P1 will push it down. So here the thrust force F2 will be pressure multiplied by the area. Let's take the area as A. Similarly, what will happen? Above it, the liquid which is present above 1, it will push it down. So here the force, let's call it as F1, pressure is P1, area A. So one force is pushing it down, one force is pushing it up. Now is there any other force which I have not drawn in the diagram? Let's quickly hear from all of you. Is there any other force which I have not drawn in the diagram? Please reply. And Chippy Pets, do we have exams after coming to the college? No, you don't have any exam beta. I said you will be directly giving NEET exam. You can directly give the NEET exam. They will be arranging special all India level NEET exam just within 2 or 3 days. You can directly give that. Rest of the students. Gravity, yes, correct. Weight, gravitational force, which is the weight. Now talking about, some are telling Sairaj buoyant force. Buoyant force beta you will study in next class. Tomorrow we will be seeing that. Buoyant force is itself due to pressure difference. Buoyant force is a force which is due to pressure difference. So the third force which is acting is nothing but the weight of the cylinder. So if we consider a liquid cylinder in between the points 1 and 2, it will have its own weight also, na? So this will be weight of the cylinder or weight of the liquid. So there are 3 forces. Now since it is in equilibrium, so net force on any such imaginary cylinders will be 0. Every portion of the liquid is in equilibrium. So downward and upward force should be balanced. So downward force, if you see, we are having F1 is acting downwards, weight of the liquid is acting downwards, and F2 is acting upwards. So these forces are balanced. So from here, if we substitute, what is F1? F1 is pressure at point 1 into area. Now what is weight of the liquid? Weight of the liquid, we know weight of the liquid is mg. Mass of the liquid, how to find mass? The density I have taken as rho. Density into volume is mass. What is volume? Area of cross section, height is h into g. So in place of weight of the liquid, I can substitute, yes, rho A hg, rho A hg. And this force is balancing F2, which is P2 into A. So from here we see, cancelling A, we get P2 minus P1 is equals to rho g h. P2 minus P1 is rho g h. That means from point 1 to point 2, if you go, that means you are going down. Okay. So if you are going down, then what does that mean? It means that the pressure is increasing. It is increasing. P2 you can see is greater than P1. Okay. When you are going up, P1 is less than P2. So we can say that as we go down, the pressure will keep on increasing inside liquid, and as we go up, the pressure will keep on decreasing. So what is the pressure variation with height? Any two points which are having a difference in level equals to h, that pressure difference will be rho g h, and always downward point or the point which is deeper, it will have the extra pressure of rho g h. Clear? Ratna is asking, sir, how mg is equals to rho g h? Mg, what is mass? Mass is density into volume. Density of the liquid is rho. What is volume of the cylinder? Area of cross section is A, height is h, A into h. So rho A h is the mass and into g. That's what we have written over here. Understood? So I think Munni, okay. So sir, pressure is a force only? No sir, no Munni, it's not a force. I think you joined the class towards the end, so you did not listen it. So in your free time, kindly see the video from the starting. You will come to know. I have explained in the beginning itself that pressure is not a force, it is an effect of a force. And which type of force? The force which exerts a thrust. Okay, thrust force, that thrust force divided by area is called pressure. So it is an effect of force, not a force. Clear? So that will be all for today. In next class we will see some questions and we will come to straight away to Archimedes principle. Okay. I hope you understood some basics which I told today. So we will be meeting again in the next class, right? Okay, thank you.
Good morning students. So today we are going to discuss about types of structural isomers or isomerism. So in last class we have studied about isomers of alkane and alkyl, that you have to remember carefully. How many students can tell about, we have written isomers of alkyl, alkane, order we have written. How many students remember for isomers of alkane order we have written in last class? Started from how many carbon atom? 4 carbon atom I have written. C4H10, C5H12, C6H14 and C7H16. You have to tell order for this. Very good. Many students are telling 2, 3, 5, 9. Very good. You have to remember this. What is sequence? 2, 3, 5 and 9. Clear? And remember, if you remember this order, then easily you can write in exam. Otherwise, it will be very difficult, because if they give C7H16, very difficult to draw 9 structures in exam. You will get 8 structures or 7 structure. So what is structural isomers? How many are possible? 9. And then we have written for alkyl group. For alkyl, from 3 carbon atoms we have started. Now C3H7, C4H9, C5H11, C6H13. What is sequence for alkyl group? Very good. Many students remember this. Very good. This is how many? 2, 4, 8, 17. 17 is less required. If you remember only 2, 4, 8, that is also sufficient for you. If you remember only this 2, 4, 8 for alkyl group, that is also sufficient. Very good. So all students, very good, you have to tell about alkane and alkyl. So this will be useful when we are practicing question for combined total number of structural isomers. So now today let us start from types of structural isomerism. Now you have to tell what is first type of structural isomer. What is first type of structural isomer? We have written last time classification or types of structural isomers or isomerism. Tell me name of first type of structural isomerism. First type of structural isomerism, what we have to discuss? Chain isomerism. What we have to discuss about? Chain isomerism. Now in isomerism, definitions are very important. So what is definition for chain isomers? If there are 2 or more compounds having same molecular formula, but their length of parent chain will be different. Their length of parent chain will be different. So they are called as which isomers? Chain isomer. But important is we cannot change functional group. Functional group will remain same. If compound is alcohol, we have to write alcohol. If compound containing 1 double bond, then another compound also having how many double bond? 1 double bond. So important is functional group must be same, but length of parent chain should be different. So those type of compounds are called as chain isomer. So what we write definition for this? That 2 or more compounds having same molecular formula, but different length of parent chain. Different length of parent chain are called as chain isomers. And this how to select parent chain, that are rules from nomenclature. Yeah. So if we take first example, let me take 4 carbon atoms. What is name of this compound? All students will tell what is name. This is normal butane. And next if I write 1, 2, 3, it says 4. Name of this compound is isobutane. I am writing common names, not IUPAC name. First compound is normal butane, second compound is isobutane. If I write molecular formula for these two, what is molecular formula for first? If you check CH3, CH2, CH2, CH3. If you write complete hydrogen also, if you write, here it is CH3. This will be CH2, CH2 and this is CH3. Same here it is CH3, CH3, CH3 and this is CH. So for first one, what is molecular formula? C4H10. And for second, what is molecular formula also? C4H10. Now if you check, these two compounds have same molecular formula, But what is different here? Length of parent chain. How many carbon atoms are present in parent chain? 4 carbon atoms. But in second one, what is length of parent chain? How many carbon atoms are there? 3 carbon atoms. So these two compounds have same molecular formula, but length of parent chain is different and both are alkanes. So it means what is relationship between these two compounds? They are chain isomers of each other. What is called? They are chain isomers of each other. If compounds are isomer, their names will be different. Here. So this is first example. Any doubt in this first example? Same molecular formula but different length of parent chain. These compounds are called as chain isomers. Any doubt in this? Clear? And let me write next example. Next example I am writing CH3, CH, CH3, CH3, C, CH3. Now let us take these two examples. Now if we write for first one, what is molecular formula for first one? How many carbon atoms are present? 1, 2, 3, 4, 5 carbon atoms. And how many hydrogen are present? 12. So what is molecular formula here? C5H12. And for second one, if we write same molecular formula C5 and H12. Now both compounds have same molecular formula. But if we select length of parent chain, what is length of parent chain here? How many carbon atoms are there? 4 carbon atoms are present in the parent chain. And in next example, how many carbon atoms are in parent chain? 3 carbon atoms are in parent chain. So having same molecular formula but different length of parent chain, so they are considered as which type of isomers? Chain isomers. And name of first one, if you write common name for this is isopentane. And common name for this structure is, what is common name? Neopentane. So they are chain isomers of each other. In first one, 4 carbon atom parent chain, next one 3 carbon atom parent chain. So first two examples we have taken for alkanes. Now we can write same examples in alkenes, alkynes, next in any functional group, any substituent. So let us take next example of any double bond, that is alkene. So if we take isobutene, next we are writing this one. Now I am not writing hydrogen. You have to tell what is molecular formula for first structure and what is molecular formula for second structure. Now what is molecular formula for first structure and what is molecular formula for second structure? Classes repeated when we came to campus. Again everything will start again from starting. Now how to write this? First, what is present here? 3 hydrogen, CH3. Here what is present? CH2. CH, here it is CH2. So how many carbon atoms are present here? 4 carbon atoms. And how many hydrogen are present? 8. And directly we can write formula. If only one double bond is present for alkenes, what is general formula? CnH2n. So if 4 carbon atoms are present, how many hydrogen will be there? 8 hydrogen atom. Clear? That we can write. This is general formula for alkenes having one double bond. Now here if we write same structure is given, means molecular formula, if you write CH3, this is also CH3, CH2, and here no hydrogen is present because for this carbon already 4 bonds are completed. So what is formula? C4 and how many hydrogen? 8 hydrogen. Now if we write, what is numbering for this? 1, 2, 3, 4. How many carbon atoms are in parent chain? 4 carbon atom. And next one, 1, 2, 3. How many carbon atoms are in parent chain? 3 carbon atoms are in parent chain. So both having same molecular formula but different length of parent chain. So that's why they are considered as which isomers? Chain isomers. And what is common name for this? It is called as butene or but1ene. If number is not mentioned, but we consider but1ene, and this is called as isobutene. This is common name, not IUPAC name. Common name butene, isobutene. So they are considered as which type of isomers? Chain isomers. Next, if we take any example of substituent, fluoro, chloro, bromo, iodo, nitro, all these are considered as substituents. So if we take CH3, CH2, 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 Cl, and here CH, see here, right, chlorine, CH3, CH3. So what do you think? What is relationship between these two? What is relation between these two compounds? Are they chain isomer or any other type of isomerism it is? It is not butene, single bond is given. In first structure, if you check what is length of parent chain, how many carbon atoms are there? 4. And in this one, how many carbon atoms are in parent chain? 3. So their length of parent chain is different. No, they are not functional isomers. They are which type of isomer? Chain. Same functional group, chlorine, chlorine, same substituent. Both are alkyl halide. Now do not consider that position of chlorine is different, 1, 2. Because in position isomerism, when we write definition for position, there we point we will write that same length of parent chain. Here length of parent chain is not same. If length of parent chain is not same, then they are considered as always which isomer? Chain isomers. Clear? Now this is example of substituent. Now let us take any example of functional group. Let us take here OH is given, and what is given here? OH. Now same functional group. Which functional group is present? OH functional group, that is alcohol. Now I am asking what is molecular formula for these two compounds? Tell me, what is molecular formula for these two? Molecular formula will be same or different? First tell me what is molecular formula. Try to find molecular formula for these two compounds. Molecular formula is same. I am asking what is molecular formula? How many carbon atoms, how many hydrogen, how many oxygen? Venugopal, oxygen is present here. You are writing C5H12. This is not alkane, alcohol functional group. There is oxygen also. OH group is present. How many? First count. How many carbon are present? 1, 2, 3, 4, 5. There is no double bond. So what is alkyl group? C5H11. And after that, which group is present? OH. So you can write this as C5H12O, or we can write C5H11OH. Same here, C5H11OH. Clear? Now if you check length of parent chain, what is length of parent chain here? 1, 2, 3, 4. And if we select length of parent chain here, 1, 2, 3. So both having same molecular formula, same functional group, but different length of parent chain. So they are considered as which type of isomers? Chain isomers. Clear? Now students, you have to tell what is common name for this first structure. Not IUPAC name, you have to tell common name. What is common name for first structure? Answer is chain isomer. Now for first structure, what we have to tell? What is common name for this? Isobutanol. No, that is not correct. Isopentanol, not correct. Common name for alcohol group is alkyl alcohol. So first we have to write name of this alkyl group. This complete alkyl group name we have to write. This complete alkyl group is isopentyl. What is name of this? Isopentyl. Total 5 carbon atoms are present. This is isopentyl. So what is IUPAC name for, or common name for this? Common name is isopentyl alcohol. For OH what we write? Alcohol. And for this alkyl group what we write? Isopentyl. And if we write common name for this, if you hide this OH, this remaining group is neopentyl. So what is name for this? Neopentyl alcohol. So these common names you can only write if you remember that class where we have written these names of alkyl group, common names of alkyl group. We have written this isopentyl, neopentyl. So isopentyl alcohol, neopentyl alcohol. These are common names. So these questions can be asked in objective, that which type of isomers are isopentyl alcohol and neopentyl alcohol. So if you know the structure, then you can tell that these are which type of isomers? Chain isomers. So this is all about which type of isomerism? Chain isomerism. If you remember definition, then you can easily tell chain isomerism. Now second type of isomers is position isomerism. Now what is definition for position isomerism? Definition is very important, that two compounds having same molecular formula. If molecular formula is same, and next important point is same length of parent chain. Parent chain should also be same, and having same functional group. Functional group will not be chain, but different position of either substituent or multiple bond or functional group. Then they are considered as which isomer? Position isomers. So if we write definition, definition is very important. Two or more compounds having same molecular formula and same length of parent chain. This is also very important, same length of parent chain, but different position of substituent or multiple bond or functional group are called as position isomers. Called as position isomers. So these two conditions we have to remember. Molecular formula is same, and next is same molecular formula and same length of parent chain. But what can be different? Position of either substituent or position of multiple bond or position of functional group. But next important point, functional group is also same. If functional group is different, then they will be called as functional isomers. So if we take example first for substituent, position of substituent. So CH3, CH2, CH2, Cl. And next is CH3, CH, Cl, CH3. Now both are 3 carbon atoms having which substituent present? Chloro. So if we take, what is length of parent chain? 3 carbon atom. Numbering is 1, 2, 3. Substituent is present at which position? First position. Now if we take parent chain, this is parent chain, length of parent chain is same 1, 2 and 3. Substituent is present at which position? Second position. If molecular mass is different, how it is possible? If molecular formula is same, how it is possible they have different molecular mass? That is not possible. There. So if there are two compounds having same length of parent chain, same molecular formula, but what is different here? Position of substituent. Substituent is present at first position, substituent is present at second position. So what is relationship between these two? They are considered as position isomers. Here. So they are considered as which type of isomers? Position isomers. Clear? So what is name for first structure? Common name you have to tell. What is common name for first structure? This Cl will be chloride. You have to tell name of this alkyl group. Not molecular formula. What is common name for this? CH3, CH2, CH2, Cl. This is normal propyl, not propane. So normal propyl, for chlorine what we write? Chloride. Alkyl chloride is common name. Chloropropane is IUPAC name. Common name is N-propyl chloride. IUPAC name easily every student can write. So they will give what their common name. So common name is what one? Normal propyl chloride. It is alkyl chloride. So name of alkyl ends with chloride. And if we write common name for this one, second structure, this is isopropyl, where hydrogen is removed from second one. So it is called as isopropyl chloride. So question will be asked, what is relationship or which type of isomers are normal propyl chloride and isopropyl chloride? So they are position isomers of each other. So this is for substituent. Next example we will write for multiple bond, where position of multiple bond will be different. So if we take example, CH3, CH2, CH double bond CH2. Next CH3, CH double bond CH, CH3. So it is very simple example. So what we have written, how many carbon atoms in chain? 4 carbon atoms. There is no branching. In second also, how many carbon atoms in chain? 4 carbon atoms. There is no branching. In first one, multiple bond is present between first and second carbon, that is first position. In second structure, multiple bond is present at second position. Why propyl, not propane? That is alkyl group. One hydrogen is removed. So these are which type of isomers? Position isomers of each other, having same length of parent chain but having different position of multiple bond. First position, and here multiple bond is present at which position? Second. So they are considered as which isomer? Position isomers. Next, if we take example for any functional group, CH3, CH, CH3, CH2, and here what is given? OH. Next is CH, now what is relationship between these two compounds? So if we select the parent chain, how many carbon atoms are present? 3 carbon atoms. If we select the parent chain, how many carbon atoms are present? 3 carbon atoms. Now if we write here 1, 2, 3. Here numbering, if we write 1, 2, 3. Now here functional group is present at which position? First position. Here functional group OH is present at which position? Second position. So what is the relationship between these two? They are considered as which isomer? Position isomers. Clear? So remember this, if position of functional group is also changed, then also they are considered as position isomer. So 3 points, what can be different? Substituent position can be different, multiple bond position can be different, or functional group position can be different. Okay. And one example in aromatic compound. So if we write aromatic benzene substituents, just remember this A. These two groups are present. Next is A. Means two substituents are present. We'll write common name for these positions. So whenever these are present at 1, 2 position, this is benzene and 2 substituents are present here. So what is given? 1, 2 substituents are present at which position? 1, 2. What is the name of second compound? Which second compound? This compound. So what is name of this? Is tertiary butyl alcohol. This is tertiary butyl alcohol. This we have written in common name of alkyl group if you check. Now move to this. If it is 1 comma 2, 1 comma 2 is called as which derivative? Ortho derivative. 1 comma 3, 1, 2, 3. Substituent is present at 1, 3. This position is considered as which position? Meta position. These are according to positions of benzene. On benzene, if substituents are 1 comma 2, that are called as ortho. If position is 1 comma 3, that is called as meta. And if position of substituent at benzene is 1 comma 4, they are called as para. So what is meaning of ortho, meta, para? Now remember, if only one substituent is present, there is no ortho, meta, para. If there are two substituents present at 1 comma 2, ortho, two substituents at 1 comma 3, meta, two substituents at 1 comma 4, para. And sometimes you will write this, here A is present, you will write this is meta, but this is nothing. There is only one substituent present. How many substituents are required? Two substituents. 1, 2 ortho, 1, 3 meta, 1, 4 para. Now if same substituent A and A are present, so what is the relationship between these compounds? They are position isomers of each other. They are which type of isomers? Position isomers. So remember this, these two substituents in every compound same. For example, chlorine, chlorine, Cl, 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 Cl. Then molecular formula will be same. Only what is different? Position of the substituent or functional group is different. So they are also considered as which isomer? Position isomers. Clear? Now in next I will write some structures. And you have to tell relationship between these two. Either they are position isomers or either they are chain isomers, or one important thing, either they are identical. If it is given, same. So write down next question. Find relationship between compounds. So only what we have to check, either chain isomer or position isomers, or they are identical. Identical is also a option. Now first part is, this is first A part, means compound A, compound B. Second structure, third structure, and this is methyl, CH3, CH3. Find relationship between these compounds. Start from first one. If you applied definition part correctly, then you will get correct answer. First one is position isomer. Somebody is saying only two options or three options. Identical is also one option. All are position isomer. No, you have to apply your mind. Think carefully about first one. First one is position isomer or chain isomer or identical. Tell me about first one only. First one is chain isomer or position. Now listen, maximum time in exam you will mark answer as position isomer. but they are not position isomer, because in carbon containing functional group parent chain start from CN, first carbon, second, third, fourth. What is length of parent chain? How many carbon atoms are there? 4 carbon atoms are present. In second one, 1, 2, 3, 4, 5. How many carbon atoms are in parent chain? 5 carbon atom. So if two compounds having same molecular formula but different length of parent chain are given, then which type of isomers they are? They are considered as which isomer? Chain isomers. So for first one, what is answer? Chain isomers. Now similar is for second part. Second part is also similar. From COH we have to start numbering carboxylic acid. So 1, 2, 3, 4. Here if you take 1, 2, 3, 4, 5. In first structure length of parent chain is how many carbon atom? 4. In second one length of parent chain is how many carbon atom? 5. So that's why they are also considered as which type of isomers? Chain isomers. So you have to apply nomenclature rules for this. For cyanide and carboxylic acid, whenever you select parent chain, first carbon atom is of functional group. Any doubt in first two part? That why they are not position isomer? Because their length of parent chain is not same. Length of parent chain is different. Now let us move to third part. Now in third part, if you write what is length of parent chain? 3, 4, 5, 6. 6 carbon atom ring. Here also 1 chloro, 1 methyl. So chloro will get priority 1, 2, 3, 4, 5, 6. Now first is length of parent chain is same. 6 carbon atom, 6 carbon atom ring. What is position of substituent here? Here position substituent is 1, 1. Here position substituent 1, 3. So same length of parent chain but different position. So that's why they are considered as which isomer? Position isomers. So in third structure, these two are position isomers of each other. Now what about fourth one? They are chain isomer, position isomers, or identical. Many students are saying identical. Very good. Now here two substituents are present. Alphabetically, which will come first, chloro or bromo? Bromo. So we have to give number 1 to this. So if you write numbering, this is 1, 2, 3. Here also if you write bromo 1, 2, 3. Now if you check same length of parent chain, same position, so it means they are identical. There is no difference between these two structures. So these two structures are identical. It means they are not isomers of each other. They are same compound. Clear? Now write down next. I'll write 2 or 3 more examples. You have to tell same, chain, position, or identical. Next, fifth example is so these three are to attempt. Last 3 examples, you have to tell chain, position, or identical. Solve these. First, fifth, you have to tell they are chain isomers or position isomers or they are identical. Fifth one, position. First select parent chain. So it is 1, 2, 3, 4. multiple bond is present at which position? First position. Here if you write 1, 2, 3, 4, position of multiple bond is also different, subsequent is also different, but length of parent chain is same. So it means they are which type of isomers? Position isomers. Now second, position isomer, chain isomers, or they are identical. For sixth one, position, chain or identical. Now you have studied this definition, then you can write. But maximum time when you see this example, you will write that these are identical. But these are not identical if you check. If you write numbering here, numbering is 1, 2, 3, 4. Here also numbering is 1, 2, 3, 4. Now subsequent position is 1, 2, 4. Here also 1, 2, 4. But what is different here? In this structure, bromo is present at which position? First position. Chloro is present at which position? Second. But if you check this structure, here chloro is present at first position and bromo is present at which position? Second position. So that's why these two are considered as which isomers? Position isomers. Overall set of locant is same. 1, 2, 4, here also 1, 2, 4. Fluorine is same, 4, 4. But position of bromine and chlorine are different. So that's why they are considered as which isomer? Position isomers. If we take last example, so CHO 1, 2, 3, 4. 4 carbon atom. Here if we take 1, 2, 3, 4, 5. 5 carbon atom. So what is the relation between these two? They are chain isomer because their length of parent chain is different. How many carbon atoms? 4 carbon atom. Here how many carbon atom? 5 carbon atoms. There. Any doubt in these examples? 5th, 6th and 7th. Position, chain. Any doubt in this? Here. So today we have covered two parts of isomerism, chain and position. Now in next class, we'll start from functional isomerism and metamerism. So functional isomerism is most important. So in next class, we'll start from functional and metamerism. So students, thank you for today.
Hello, good morning, students. Good morning. Good morning, all of you. Good morning. Good morning. Good morning. Yes, yes, good morning. How are you all, guys? When will the offline class starts? When it starts, you will come to know, no? Yeah, I'm fine. I'm good. I missed you. Wow, butterfly. Even me too. Good. Enjoying your stay at home, I guess. Yeah, fine. Okay. I'm good. Do you remember which topic I had dealt previously? Do you remember? Miss, oh, thank you so much. Okay, you don't remember? Okay. I'll tell you it is cell. Cell chapter we have completed, like cell organelles. Yes. Very good. Seepana. Okay, Tikshana. Yes, cell organelles. Very good. Cell. Do you want me to ask some questions? Yeah, chromosome also is there. Very good. Some questions? 2-3 questions? Very good. Rockstar. Okay. Sindhu. Yes. Nikhil. Vishnu. Cell. Wow, good. Okay. I'll ask one simple question which you can tell me. Okay. Which are the powerhouses of this cell? Which cell organelles are called as powerhouses of this cell? Yeah, good morning. What is another name of cell membrane? Plasma membrane. Mitochondria. Very good. So all my students are very active now. Good. Mitochondria. Very good. Okay, then one more question. One more. Very good. Keep it up. Which cell organelles are present in all the living organisms? Which cell organelles are present in all the living organisms? Options are mitochondria, chloroplasts, ribosomes. Butterfly, very good. Ribosomes. Sairaj, wow, good. Not cytoplasm, ribosomes. Very good. Ribosomes. Good. Okay. Next question. Very good. Name the double membranous enveloped cell organelles. Name the double membranous cell organelles. Mitochondria, correct, Ishant. Nucleus, okay, but ribosomes, they don't have. Mitochondria, correct. Very good, mitochondria. Very good, Eshu. Yes, Eshu, chloroplast. Mitochondria, chloroplast and nucleus. 3 are there, mitochondria, chloroplast and nucleus. Very good. Chloroplast also, yes. 3 are there. Mitochondria, chloroplast and nucleus. All these 3 are having 2 membranes. Okay, last question of the previous topic. Name the amembranous cell organelles. Amembranous cell organelles of living organisms. Amembranous. Very good, Tikshana. Ribosomes. Yes. Ribosomes. And one more is there. Lysosomes. No, butterfly. Lysosomes will have one membrane. Ribosomes, correct. There is one more. In animal cells it is present. Only in animal cells. Centrosomes. Very good. Centrosomes. Correct. Yes. Ribosomes and centrosomes. Good. Okay. Flagella, it is covered by plasma membrane, but it is not a cell organelle, no, it is an surface structure. It is outermost surface structure of the cell. So, ribosomes and centrosomes. Very good, very good. Good. Okay. Now, which is called as kitchen of the cell? Kitchen of the cell. Which cell organelle is called as kitchen? Where we'll be cooking so many things, no? Which cell organelle is called as kitchen of the cell? Very good, butterfly. Chloroplast, yes. Koteswar Rao, yes. Chloroplast, yes, yes. Not mitochondria, not ribosomes. Where food is synthesized? Chloroplast. Correct. Okay, very good. Which is considered as the brain of the cell? Brain or master of the cell? Yeah, chloroplast is correct. Which is considered as the brain of the cell? Very good, Sairaj. Nucleus. Very good. Varshita. Very good. Yes, yes. Balu. Yes. All of you are correct. Nucleus. So, I hope you have completed the NCERT cell chapter. Yes. Read that NCERT. If you have that book. Yes, nucleus. Very good, Amma. Very good. Let me now proceed to the actual topic of today's. That is cell cycle. All of you can see on the board cell cycle. Okay. Cell cycle, in this word, the cell is undergoing cyclical events. That means origin point, the starting point, starting and ending. Both points are same. So, anything if it is happening in a cycle or round manner, where the starting point and ending point are same, that is considered as a cycle. So, now cell is undergoing the cycle of events. So many cyclic events will be happening in the cell. That is called as cell cycle. In this cycle of events, what will happen is, it is a sequence of events in which, first thing, duplication of genome will happen. Duplication of genome. It is nothing but DNA. You will be understanding the genome means like a total set of chromosomes, but here you all consider DNA. You know, duplication means if I have one DNA, it will become two DNAs. That is called as duplication. Duplication of the genome. Okay. That is the main thing. It involves the replication of DNA. And then it involves the cell growth. Cell will be growing. Then it involves the cell division. So, the cyclical events or sequence of events which include duplication of genome, then cell growth and then cell division. So, first duplication will be happening along with the cell growth, and after that the cell will be dividing into two daughter cells. So, all these cyclical events will be together called as cell cycle. Among all these, the important step is the cell division. If I give you one cell, one parental cell, this is called as one parental cell. Okay, this has to undergo all the sequence of events like duplication of DNA. Here the DNA will be there, which is duplicated like this. And later it will increase in its size, it will become big with all the chromosomes. Okay, like this, it will become big, cell growth, and later this one will divide into two daughter cells. Two daughter cells. So, starting from here, the cell will be growing and it will be duplicating the genome in terms of DNA replication, and then the cell will be dividing into two daughter cells, is called as cell cycle. So, one cell will be entering here and finally you will be getting two daughter cells. So, cyclical events will be happening in the cell, and by the end of this you will be getting the two daughter cells. That is called as cell cycle. Okay? Is that clear for everyone? So, the sequence of events like the duplication of genome in terms of DNA replication, then cell growth and cell division. All these three events together, it is called as cell cycle. Understood all of you? Is that clear? Yes? Okay, very good. So now let us see what is going to happen actually in the cell cycle. Okay, all of you see here, this is the cell cycle. Okay, in this, all of you can see very clearly that the first one, we have to start at this point, okay, and it will be going like this. This is the starting point, first one. And cell cycle is actually divided into two phases. One is interphase and the second one is M phase or mitotic phase. So in the cell cycle, we have two phases. One is interphase. The first one is interphase and the second one is the M phase or mitotic phase. Now let us see in this, cell cycle is divided into two phases. One is interphase and another one is M phase, which is also called as mitotic phase. So this interphase, okay, is further subdivided into G1. You all can see here G1, then S phase, then G2 phases. So interphase is further divided into, this is further divided into G1 phase, S phase and G2 phase. G1 is called as first gap phase, or you can call it as first growth phase. S phase is called synthesis phase. G2 is called second gap phase or second growth phase. So interphase is subdivided into three phases, G1, S and G2. This is the sequence. Cell cycle is divided into two phases. First one is interphase, second one is M phase. In the M phase, actual division will happen. So it is also called divisional phase. Divisional phase. So M phase is also called as divisional phase. So in any cell cycle, you can take any cell, okay, which is undergoing the division. So in that cell division, which is a part of cell cycle, it includes two phases, interphase and M phase, and interphase is further subdivided into G1, S and G2. M phase is also called as divisional phase because in that phase only the actual cell division occurs and one cell will be dividing into two daughter cells. And M stands for mitotic phase. Now, you know the subphases of interphase G1, S and G2. G1 is called as first gap or first growth phase. S phase name is synthesis phase and G2 is a second gap or second growth phase. What are the events that will happen in the interphase? Today let us see the interphase only, then we will proceed to the M phase mitotic phase in the next class tomorrow. Okay, all of you clear? Prophase, metaphase, yes, anaphase and telophase. Okay, cell cycle is divided into how many phases? Cell cycle is divided into how many phases? How many phases? 1, 2, 3, 4. How many? How many? 2. Sairaj, very good, 2. 2. Most attentive person. Yeah, Shrestha, okay. Good. 2 phases. The first one is called, first one name. First one. Yeah, 2 correct. Which is the first phase? Interphase, Tikshana, very good, keep it up. Interphase. Second phase is called, second phase. First one is interphase, yes. Second one. Second one. M phase, yes, M phase, very good. M phase is also called as divisional phase. Yes, very good. Now all of you, I did not tell you this point. Okay, if you are clever enough, you will answer to this question. Focus on the diagram. Okay, focus on the diagram. Now my question is, okay, are you all ready? Ready? I did not tell you this, but I am asking one question which is there in this diagram only. Ready? Yes, okay. In the cell cycle, okay, which is taking longer time? Which is taking longer time? We have two phases in the cell cycle, which is taking longer time. All of you check properly. The answer is here only. Yes, Sairaj, very good, very good. Interphase, yes. Don't say G1 immediately. Yeah, I have asked only phase, not subphases. These three are called as subphases. Yeah, G1 is correct, but it is a subphase. The question is about phase. See all of you here. These are phases. These are subphases. So my question is about which is taking longer time, which phase is taking longer time. So interphase. G1 is answer for which subphase or which phase of interphase is taking the longer time. So for my question, the answer is interphase only. Have all understood? Which phase is taking more time for the completion? It is interphase. In interphase, which is taking longer time? Yeah, G1 is correct. All of you can see here, the green color one is given more time. Okay. So you can see very clearly that G1, it is taking this much time. So first longest phase in the interphase, then S phase, second one, third one, G2. That is the order which you have seen, but then the G2, definitely S phase is longer, but in the diagram it is looking more or less same. So first one is G1, it will take longer time, then S phase and then G2 phase. But can you all see one thing, it is there outside this G0. Can you see G0? Yes, G0, can you all see? It is given as resting. Some cells, okay, without entering into the interphase, they will come out from the G1. From G1, they'll come out and they'll be resting. That resting phase is called as G0. It is also called quiescent stage, where the cells will be active, but they will not divide. They'll be resting and they'll be active, but they do not divide. They'll do all the metabolic activities, but they won't divide. For example, our heart cells and our nerve cells, they have already stopped the division. So they will enter into the period of rest, which is called as G0 phase, or you can call it as quiescent stage. Quiescent stage or quiescent phase. Only some cells will be entering temporarily or permanently into the G0 phase, where they'll be taking rest, but it is metabolically active phase. So some cells like heart cells, nerve cells, okay, they will enter into the permanent arrest of cell division. So they will be entering into the G0 permanently. But there are some plant cells like parenchyma, collenchyma, they will enter temporarily into the G0 phase, where they'll be arresting the cell division for some time and later they resume the division whenever the plant needs. Then they will resume their division. So that is the G0 phase, which is also called as resting phase, which is metabolically active, and it is also called as quiescent stage. Okay. If the cell is entering into the G0, it will enter from which phase? Is it entering from G1 or S phase or G2? From where the cell is entering into the G0? From which phase the cells are entering into the G0? You can see in the diagram only. Yes. Very good, Jayashree. Very good, Sairaj. Sairaj, MM from G1. From G1, all of you can see clearly. From G1, this is entering here. So the cell is entering from G1 to G0 to stop the division. If not, if it is not going to G0, then obviously from G1 the cell will enter into S phase, and from there the cell will enter into G2 phase. Okay. Generally, the cells will be coming out of G1 if they want to stop the cell division. Okay. If not, they'll continue the cell division in the interphase, that is G1, afterwards S phase, afterwards G2. Okay. I hope all of you understood. Okay. Which is the, okay, how many phases are there in the interphase? How many phases? How many phases are there? How many phases are there in the interphase? 3. Very good. What are they in the sequence? In interphase I am asking. In interphase. 3. 3 is the answer. Not 4, ma. Someone type 4. 3. In interphase only 3 subphases are there. They are in the order S butterfly, G1, S and G2. Very good. G1. What is the sequence of the subphases of interphase? G1 first. S second one. Okay. Then third one is the G2. It will proceed in this manner. The interphase will be happening in the sequence. It will start with G1, then S phase, then G2 phase. Okay. Now for example, I am taking one yeast cell. Yeast is a fungus. You know, it is a unicellular, one cell. You must have got in your school. The yeast will be looking like this. Right? So yeast is the unicellular edible fungus. We can eat that because it is used in the bakery items, bakery industry to prepare bread and all. So unicellular, it is a fungus, edible fungus basically, and it will take to complete the total cell cycle. Total cell cycle takes 90 minutes. Cell cycle time period. 90 minutes. Okay. If you take the human cell, so for completing cell cycle, it will take 24 hours. For the total cell cycle, the human cell will be taking 24 hours, 1 day, and yeast cell will be taking to complete the total cell cycle, that is interphase and M phase, to complete the total cell cycle, it will take 90 minutes. Okay, 24 hours human cell. And yeast cell will be taking the 90 minutes, 1.5 hour. 1.5 hour. So, in this total time period of cell cycle, the 24 hours or the 90 minutes, okay, the interphase will be taking more than 95%. Interphase will take more than 95% of time in the cell cycle. So, for example, if you take the human cell, you know 24 hours is the total cell cycle time period. More than 95% of the time means 23 hours or more than 23 hours. It will be taking for interphase. This is taking in cell cycle, interphase will be taking 23 hours, whereas M phase will be taking just 1 hour. So, less than 5% will be given to the M phase, the actual division. So, more than 95% of the time of the cell cycle will be given to the interphase. It will be going for the interphase like G1, S and G2. So, most of the time it will be spending in the interphase, and just less than 5% of the time it will be using for the actual division, that is M phase. So, if you take the human cell of 24 hour cell division, then interphase will be using 23 hours and M phase will be using only 1 hour for the human cell. Then can you calculate for this also how much time it will be taking? More than 95%. In 90 minutes, more than 90% will be given to the interphase and less than 5% will be given to the M phase. So, most of the time in the cell cycle will be involving interphase. In that also particularly G1 phase first. Then it will be the S phase and then it will be the G2 phase. All of you can see very clearly in this diagram. Now, the first phase, you know, G1 is the first phase and it is called as first growth phase or first gap phase. First growth or first gap phase. Here in the G1, most of the cell organelles, RNA, proteins, most of cell organelles will be multiplied. Okay, like lysosomes, ribosomes, all of them will be multiplied here. So, all the accumulation of cell organelles, okay, then proteins, different types of proteins accumulation, then RNA synthesis, everything will be happening in the G1. Even energy, ATP energy also will be synthesized in the G1, which is required for the cell division. Interphase is actually, initially it was called as resting phase. But now we understood that in interphase, the cell is preparing itself to undergo the division. It is preparing so many materials. Now that you can see in the G1, it is preparing to synthesize the RNA, it is preparing proteins, it is preparing cell organelles because it has to distribute to the daughter cells. So everything preparation is done in the interphase. Though interphase is resting phase, it is metabolically doing all the preparation to undergo the cell division. So here in G1, RNA synthesis, protein synthesis and most of the cell organelle synthesis is happening, which is increasing the cell growth. Cytoplasm contents are increasing, nuclear contents are increasing. So the cell is growing in its size and it is taking more time out of the three. G1 is taking more time. Then after this, the cell growth will be a continuous process which will be happening even in the S phase continuously, but you cannot see that much accumulation. So it started with cell growth initially and where RNA, protein, cell organelles got accumulated in the nucleus and in the cytoplasm. So cell is becoming big. Then DNA synthesis. As the DNA is synthesized, S phase is called as DNA synthesis synthetic phase. S stands for synthesis. So synthesis means what it is synthesizing here, not RNA, not proteins. It is synthesizing mainly DNA. DNA is actually undergoing replication. That means one DNA, if it is there, it will become two daughter DNAs. So the DNA replication occurs in the S phase. So one DNA, if you take here, it will become two DNAs, two daughter DNAs. Imagine a nucleus has 2C amount of DNA. Imagine 2C. I am giving it just a number. DNA content is always measured in terms of C values. So I am taking in G1, it has the 2C amount of DNA. So the cell, when it enters into the S phase, in S phase, after the replication of DNA, in S phase, the DNA would have become 4C. So before, one DNA is there with 2C amount. Now here the DNA got replicated, so it means it got xeroxed. One more DNA got formed. So the DNA content increased to 4C. Always a cell, whatever it is having in the G1 phase, DNA, whatever, if you want you can take C or 4C, whatever. Generally we will say 2C is present in the G1. So that 2C will become double in the S phase. By the end of the S phase, you will have double the amount of DNA because the DNA is undergoing replication here. Okay, that is the main event of synthetic phase, DNA synthesis. It is happening by DNA replication, which will be done in the nucleus because DNA is present in the chromosomes. This is the first event. The next event what will happen is in the cytoplasm, if it is an animal cell, only in animal cells, centrioles will be duplicated. Centriole duplication. Centrioles duplication occurs in the cytoplasm. This occurs in the nucleus, this occurs in the cytoplasm. So, two events are made in the S phase, that is, one is DNA replication, one DNA is becoming two daughter DNAs. 2C amount of DNA of G1 is becoming the 4C amount of DNA. And the second one is centrioles duplication. Two centrioles will be there, you have learnt in the animal cell. They are present near the nucleus and all. The two centrioles will be duplicated and they will become 4 centrioles in the cytoplasm. So, these two are the events of the S phase, synthesis phase. So, DNA replication and centrioles duplication if it is animal cells only. This is the event. But remaining all cells, this event is common, DNA replication. Only in the animal cells you have one more extra role in the S phase, that is the centriole duplication. So, in S phase, DNA synthesis and centrioles are also duplicated. Then the cell will be entering into the third phase, particularly in the interphase, that is G2. G2 means second growth or second gap phase. Second growth or second gap phase. In that also cell growth will be continued. Cell growth will be continued. Here also RNA will be synthesized, proteins will be synthesized, okay, particularly the proteins like tubulin. Tubulin is forming the microtubules and microtubules will be forming the spindle fibers because the cell has to undergo the cell division. So, particular proteins will be there like tubulin proteins that will be synthesized here in G2, okay. And then in G2, other cell organelles which can't be replicated or duplicated in the G1 like mitochondria, Golgi, ER, they will be multiplied here. Here except mitochondria, Golgi and ER, remaining all cell organelles will be multiplied here. In G2, mitochondria, ER and Golgi complex will be multiplied. That means few cell organelles will be multiplied here, duplicated here. Here most of the cell organelles will be multiplied or duplicated here. They will be forming different cell organelles for the equal distribution. So, cell growth is happening continuously from G1 to G2. It begins with G1 and it ends with the G2. In G1, RNA, protein synthesis and most of the cell organelles are being duplicated or replicated. Then in the S phase, DNA is undergoing the replication. One DNA is becoming two daughter DNAs. Then centrioles duplication will happen in the cytoplasm if it is an animal cell. Then in G2, cell growth will be continued where RNA and tubulin proteins will be formed which are necessary for cell division in the future. And then cell organelles like mitochondria, Golgi and ER also will be duplicated here. That means the cell has prepared everything what is required for cell division by the end of the interphase. Then now the cell will be entering into the M phase, mitosis. M phase is mitotic phase or divisional phase. That M phase, all of you can see, it is happening here by karyokinesis. The first one is karyokinesis and second one is cytokinesis. By the end of this, you will be getting two daughter cells are formed. Okay. So, M phase will be nothing but the divisional phase where first the cell will be entering into the karyokinesis and then cytokinesis. Let us see the meaning of the word karyokinesis and cytokinesis. Yeah, all of you can see here clearly. The first one, you know that cells are maturing G1. If they mature, they will enter into the G0. The cell cycle is stopped, halted. Halted means stopped. Otherwise, the cell will be continuing the cell division. From G1, it enters into the S phase where the DNA is copied. Then the cell division will be continued. The cell prepares for division. This is G2, which is also cell growth. After that, the cell is entering into the M phase or mitotic phase where you have these four names which you already know: prophase, metaphase, anaphase, telophase. These four will be coming under nuclear division. And the last one is cytoplasmic division. By the end of this one cell, it has become the two daughter cells. So, how many daughter cells are formed ultimately at the end of the cell cycle? By the mitosis, you will be getting the two daughter cells. So, M phase is divided into karyokinesis and cytokinesis. After the interphase is done, the cell will be entering into the M phase. That M phase is divided into karyokinesis and cytokinesis. Karyo refers to nucleus, nuclear. Kinesis refers to division. So, in the karyokinesis, nucleus of one cell will divide into two daughter nuclei. Cytokinesis means cytoplasmic division. Cytoplasm will divide into two and it will be dividing one cell into two daughter cells. One cell will be dividing into two daughter cells by the end of the cytokinesis. So, M phase, first nuclear division will happen, one nucleus will become two daughter nuclei. Then the cytoplasm will divide them to form the two daughter cells. For example, all of you can see this is the one cell, okay. Then first the nuclei will divide, two nuclei, and then this will divide into two daughter cells. Two nuclei are first to form karyokinesis and then a single parent cell divides into two daughter cells. So, this after completion, the daughter cells can continue the division. They can continue the division in the G1 or they can exit the cell cycle. They can come out depending on the cell type, heart cells, nerve cells, like that. They will come out of the cell division permanently and some cells will be arresting the cell division temporarily. So, they will enter into the G0 either temporarily or permanently. This is how the cell cycle will be happening. In the human cell, it will be taking 24 hours, where 23 hours is there for G1, S and G2 and 1 hour is there for M phase, that is for karyokinesis and cytokinesis. I hope all of you understood this. Yes, guys? Yes, any doubts? Any doubts? Any doubts? Any doubts? 60. What is that? 60 included in interphase? G0. Okay, sorry. G0 is not in interphase, it will come out of that. It will come out. Cell cycle includes only interphase and M phase. G0 is arresting the cell division. When the cell cycle is stopped, they will enter into the G0. Okay. Clear? Okay. G0 is not included in the interphase. G0, it is not there in the interphase. From the interphase, they will come out into G0. They can't come out from the M phase. They will enter into G1 first, and if the cell wants to stop the division, so it will come out from G1 to G0. Where the G0 is a quiescent stage where the cell division cannot be continued, it will be arresting the cell division. It is also called quiescent stage. It is metabolically active stage. Yeah. Okay. Once again, mitotic phase. Okay. Mitotic phase is divided into two phases, karyokinesis and cytokinesis. Karyokinesis means nuclear division. Cytokinesis means cytoplasmic division. One cell will be having one nucleus, you have learnt. Some cells may have two nuclei also. That nucleus first it will divide into two nuclei and then the cytoplasm will be dividing exactly in the middle like this and the parent cell will be dividing into two daughter cells. About all the details of karyokinesis and all you will be learning tomorrow. M phase, you know, you have learnt prophase, metaphase, anaphase and telophase. They are the four phases of the new karyokinesis that you will be learning tomorrow. I am not talking about M phase today, just the brief introduction. Okay, M phase we will see tomorrow, that is mitosis. Okay. Have all understood up to here? Doubt. What is it, Mallikarjuna? What is it? Yes, okay. What is no, ma'am? Himaja. When we are hurt, then we use what is that? Creams. How does it work? Okay. See, that is cell repair. They will be doing the proliferation. They will undergo the mitosis only, and the damaged cells are replaced by the mitotic cell division. It will be filling up the new cells there. All the damaged cells will be removed, and in that place new cells will be formed there. That's how the creams, that Mederma, whatever the creams are there, they'll be working out. It is because of mitosis. Basically, the process involved in the cell repair is mitosis. That you will be learning tomorrow. Okay? Clear? I hope all of you understood. Okay. Now let us see the next one. Here I have given you the highlights of the cell cycle. In this you can see why I have given you one more time this G1, S and G2 means, because I wanted to show you checkpoints. There are certain points where we can arrest the cell division by using certain antibiotics, okay, like that we can arrest the cell division. So once the cell crosses certain points, then it has to undergo the division, otherwise it can stop. I told you from G1 it will enter into G0, right? Permanent or temporary arrest of cell division. Now let us see here where are the checkpoints are present. In this, all of you can see G1 and G0, G1 and S checkpoint. This checkpoint is here, G1 and S checkpoint. This checkpoint is checking for nutrients, growth factors and DNA damage. If any nutrients are not there, if the growth is not happening fully, if the DNA got damaged and all, the cell will not cross the checkpoint. G1 and S checkpoint is called as major checkpoint. So once the cell crosses this, there is no stopping of cell division. There is no stopping of cell division if it crosses the first checkpoint, that is G1 and S. Then second checkpoint, all of you can see G2 checkpoint here. The cell size, and it will check for DNA replication. If the cell has reached the maximum size or not, and DNA replication has been completed or not. But there is no stopping of cell division here. It will come back and it will do the replication of DNA. That process will be completed and it will resume the division. It cannot stop the cell division once the checkpoint of G1 and S it is crossed. It has to undergo the division. Then metaphase checkpoint. You know, in the karyokinesis we have prophase, metaphase, anaphase and telophase. Tomorrow you will get that. So metaphase checkpoint is there. It will check for spindle attachment. Whether all the, I have told you in the chromosome structure, spindle fibers will come and attach to kinetochores. Centrosome is there, centromere is there. That centromere on both the sides, some small discs will be there. For that, spindle fibers will come and attach. Okay, that I will tell you tomorrow. So that checkpoint is there, but it will check for that whether chromosomes spindle attachment happened or not, and again it will continue the division. There is no stopping of cell division if the cell crosses this G1 and S checkpoint. So there are basically three checkpoints are there. First one, G1 and S checkpoint. This is called major checkpoint. Once this crosses, there is no stopping of cell division. Then G2 checkpoint, it will check for replication and it will continue the division. Then M checkpoint, metaphase checkpoint is there, where chromosomes are attached to the spindle fibers or not, it will be checked and the cell will continue the division to form the two daughter cells. This is about checkpoints. So we have three checkpoints. Three checkpoints are there. First one, G1 and S checkpoint. Second one, G2 checkpoint. G2M it is called as. And third checkpoint is called metaphase checkpoint. But once the cell crosses this, after first, the cell has to continue the division. There is no stopping of the cell division. Once it crosses the G1S checkpoint, then it has to continue the division by rectifying all the mistakes. I hope all of you understood. Yes? Yes, students? Yes, yes, yes. Okay. So, all of you are going to prepare this. Okay. Interface, particularly cell cycle and interface. And come tomorrow for the class to answer the questions. Any doubts? Any doubts? When the college starts, you will come to know. They'll inform you well in advance. In fact, than us, you will be knowing first. No doubts. Okay, good, good, good. Okay, then. Okay. Okay, see you guys tomorrow. Thank you.
So good morning, students. Hello there. Good morning, students. How are you? Yes, very good morning. Yeah, I'm good. Yeah, sure. Good, good. Fine, very good. Rishita. Very nice. How you doing? All fine? All's good? Yes, Akhil. When is the last online class? You'll be informed. Don't worry. Don't worry. Don't worry. Do not keep on repeating same sentence. That's okay. You'll get answer. Okay. So last class we discussed about ultrastructure of a striated skeletal muscle. We learnt about actin thin filaments, myosin thick filaments, isotropic bands, anisotropic bands, Z lines, M lines. As I told you already, because of arrangement of actin and myosin attached to Z line and M line respectively, we were able to see a pattern which is visible under microscope like striations. I bands, A bands, I bands, A bands, thin thick filaments, isn't it? That gave us striations to muscle. Okay. Today we will be slightly partially learning about how does muscle contract, relax, and then we can also learn about human skeletal system. Okay, so just basic fundamental aspect for you today. Z line, M line. Guys, Z line is elastic, M line is fibrous, right? Question for you. Z line was attached to thin filaments or thick filaments? Z line is attached to thin filaments or thick filaments? Answer this. Very good. Z line is attached to thin filaments, whereas M line was attached to thick filaments. Okay, I will show you half of this. So thin filaments, right? I can show on both sides, that's okay. Done? Now M line was attached to thick filaments, right? Just a little improvement. Okay. Now, as I told you, thin filament has actin, actin has got myosin binding site. Thick filament is myosin, myosin has got actin binding site, which means thin filaments can attach to thick filaments and thick filaments vice versa can also attach to thin filaments, right? So how does muscle do this? Whenever there was a nerve impulse, I mean a response to contract a muscle, a chemical signal, this chemical signal reaches sarcolemma, plasma membrane of muscle cell,
and from plasma lemma the impulse will be given to sarcoplasmic reticulum. Okay, so nervous system passes on signal to sarcolemma. Sarcolemma passes the signal to sarcoplasmic reticulum. And because of this signal, sarcoplasmic reticulum releases out calcium into sarcoplasm. Okay, so nerve impulse touches sarcolemma, sarcolemma send impulses, sarcolemma activates sarcoplasmic reticulum, and now this sarcoplasmic reticulum release calcium ions into sarcoplasm. These calcium ions of sarcoplasmic reticulum, they come and attach to actin, that is thin filaments. So calcium bonds to or binds to which filament? Thin filament or thick filament? Only thin filaments. See this? So now this calcium, it binds to thin filament, nothing but actin. Okay, see, nervous system to sarcolemma, sarcoplasmic reticulum, calcium into sarcoplasm, and sarcoplasm has calciums, calcium attaches to thin filaments. Okay, because of this attachment of calcium to thin filaments, now actin and myosin will get an opportunity to bind with each other like this. Now actin and myosin will get an opportunity to bind like this. Yeah. See? See guys, actin was attached to myosin. Actin was inside thin filaments. Myosin was inside thick filaments, right? Done? See guys, because of calcium, actin will bind to myosin. And now myosin will recruit ATP. Myosin will recruit ATP and pulls entire actin towards it like this. I'm telling again. So for you, I'm showing a graphic like this. Please focus here. This is Z line, right? Attached to actins. Now this is M line, which is attached to myosins, thick filaments. See this carefully to understand in simple language. Keep your language very simple. Done? Clear, guys? Visible? Z line, M line, right? So actins were bound to calciums, and myosin has recruited ATP. Myosin has recruited, it took help from ATP, and actin took help from calciums, right? Once they attach with each other like this, okay, or I can show you like this. This is myosin, this is actin. So myosin will try to pull actin like this. So myosin, with the help of ATP, will try to pull actin like this. See this? If it pulls like this, see this clearly. Myosin is pulling actin, contraction. Myosin has released actin, relaxation. Myosin pulled actin, contraction. Myosin released actin, relaxation. Thick filaments pull, thin filaments, contraction. Thick filaments leave thin filaments, relaxation. M line is pulling Z line, contraction. M line has left Z line, relaxation. See, understood? So thin filaments were sliding on thick filaments only, isn't it? You can see, guys, thin filaments were sliding, not thick filaments. Thin filaments were sliding on thick filaments. And this theory of muscle contraction was referred as sliding filament theory. Once your offline classes start, we can go into details, exactly what exactly happened. See, again, this is contracted muscle, right? If there is no ATP, relaxed. Sir Garuda, sir, what will contract and relax? So if thin filaments are sliding towards, if thin filaments are going inward like this, entire muscle cell size decreases. Decrease in size is considered as contraction. Because of sliding of thin filaments on thick filaments, overall muscle gets contracted. And this contraction is aided by two chemicals. Calciums attached to actin and ATP associated with myosin. Because of calciums and myosin, there is muscle contraction. Done. I'm giving you basic view only, outline of this theory. I'm not getting into details completely. Okay. Can see, this is Z line, right? Z line is moving towards M line, isn't it? Relaxation. So moving towards M line, contraction, coming back, relaxation. Actin thin filaments are sliding on thick filaments, contraction, going back, relaxation, like this. Understood? Basic aspect. So it is fundamentally thin filaments are sliding on thick filaments. This simple theory was referred as sliding filament theory. It has only one postulate for you: thin filaments, that is actin, slide on thick filaments. If they slide inward towards M line, that is contraction. If they slide outward, go back to normal state, that is relaxation. So contraction, relaxation, contraction, relaxation. Beta, contraction is decrease in size of muscle. Relaxation was muscle coming back to its original shape position again. So contraction, relaxation, contraction, relaxation. Contraction is because of sliding of thin filaments on thick filaments. Relaxation, they get back again. Contraction, relaxation, contraction, relaxation. Understood? This is clear, basic concept. And this contraction requires ATP. Therefore, contraction is obviously active process. Relaxation does not require any ATP. Therefore, relaxation was passive process. Isn't it? Guys, contraction requires ATP. Relaxation does not require any ATP. This point we also discussed in respiration. Inhalation is contraction, exhalation is relaxation. Inhalation requires ATP, active. Exhalation does not require ATP, passive. And at that location, which muscle is contracting? Intercostal muscles as well as diaphragm also during inhalation. Very good, Tagore. Both intercostal muscles and diaphragm, both are exhibiting contraction during inhalation, and in relaxation, both intercostals as well as diaphragm gets relaxed. Therefore, inhalation is contraction, active, exhalation is relaxation, passive. Same rule was applicable in every location. Inhalation, exhalation. Inhalation, exhalation. Inhalation, exhalation, like this. Done? Where does ATP come from? ATP was already inside cytoplasm because cell was performing glycolysis. Isn't it? ATP will be provided. Why there was mitochondria? Mitochondria main role is to manufacture ATP for cell metabolism. Okay? Done with this? Yeah. Now, the entire muscular system was not attached to the skeleton. Only a part of muscle was attached to the skeleton. The muscle which was attached to skeleton was skeletal voluntary muscle, right? Muscle to bone with the help of tendons. So the only muscle which is attached to bone was skeleton was voluntary skeletal muscle. And the question was, guys, when you speak about human skeletal system, even human skeleton was also derived from same embryonic layer from which you got muscle. So among, you have got a question now. Which embryonic layer does give muscle? Mesoderm. Which embryonic layer gives blood? Mesoderm. Which embryonic layer gives connective tissues? Mesoderm. Which embryonic layer gives skeletal system, bones? Mesoderm again. Yes, 99% of connective tissue systems, supporting systems come from mesoderm. Means we are triploblastic in nature. It comes from mesoderm. Bones come from mesoderm. Okay, so human skeletal system. When you are born, in fetal condition at least you may have 280 to 300 bones. Yes, that was there. But while you grow up, some bones fuse with each other, and the final count of bones in adult was only 206. Yes. Now question. Number of bones in fetal condition is more than number of bones in adult. Yes. What was the reason? Some bones fuse with each other. There was fusion. Therefore, obviously, number of bones may decrease. Guys, for our better understanding, we can actually divide human skeletal system. Skeletal system, it's up to you. You can write skeleton or skeletal, it's up to you, no issues with that. Could be split into two parts. Just for bookish purpose, learning purpose, there was no splitting actually inside a body. So for our convenience, we will be learning human skeleton in two categories. All those bones found along your body axis. The same axis was used. We call it as sagittal axis, which was used to cut your body into two lateral halves. The axis which gave you bilateral symmetry, same axis, like this. All those bones found along this axis. So all those bones found along this axis, it represents axial skeleton. And guys, axial skeleton consists of only 80 bones. Whereas your limbs, forelimbs, hind limbs, 120 bones were there. And your limbs were attached to your main axial skeleton with the help of girdles, pectoral girdle and pelvic girdle. This is shoulder, it is hip. So all those bones found in your appendages and girdles, those are almost 126 bones, and they represent your appendicular skeleton. So human skeleton could be split into two parts, axial and appendicular. Axial is having 80 bones and appendicular skeleton consists of maximum 126 bones. So total was around 206 again. Now let me, guys, please help me to count bones in axial skeleton. Shall we count them? Okay. Axial. So what were in axial skeleton? So bones in your head region, done, entire head, bone, front, sternum, and bones attached to sternum. What are those? Ribs. And we are able to stand, sit, run, walk, jump and swim because of this backbone or spine. What do you call this? Vertebral column. Vertebral column. Synonymously, spine, because it protects spinal cord. Now in head region also, there were 8 bones of your skull. Okay, so skull was made up of 8 bones. There were 14 bones in your face. These are called as facial bones. Therefore, facial bones are 14. Some books mention skull bones as cranial bones. Accepted. So cranial bones 8, facial bones 14, and a bone at the base of your tongue. Tongue was attached to a U-shaped bone called as hyoid. So write it also, hyoid bone. Only one tongue, therefore one bone. So what was number till now? 8 plus 14 plus 1, right? 23. And there are 6 bones, 3 each side, 3 here, 3 here, which help you to listen. We call them as ear ossicles, malleus, incus, stapes. Malleus, incus, stapes. Outermost malleus, middle incus, innermost stapes. Stapes was smallest bone of your body. Among 206 bones, 2 are smallest. What are those? Stapes. We have 2 stapes. Malleus, incus, stapes. These 6 bones were called as ear ossicles. Ear ossicles. How many are there total? 6. So your head has almost 29 bones. Done? And sternum is a single bone. T-shaped bone, sternum. Correct? Then ribs, you have got 12 pairs of ribs. 12 pairs means 24 bones. Guys, ribs, 12 pairs. Among 12 pairs of ribs, 7 pairs of ribs were attached to both sternum and vertebrae. We call them as vertebro-sternal ribs. How many are there? 7 pairs, 14 ribs only. 8, 9, 10, 11, 12 were attached to vertebrae. 8, 9, 10 was not directly attached to sternum, and 11, 12 were never attached to sternum. Therefore, first 7 pairs could be referred as true ribs. 8, 9, 10, 11, 12 could be referred as false ribs. Okay, so we have 24 ribs. Done? And vertebrae. Vertebrae. Number of vertebrae in your neck region, we call them as cervical vertebrae, 7. 7 neck vertebrae. Number of vertebrae in your thoracic region, how many are there? 12 thoracic vertebrae. Number of vertebrae in your abdominal stomach region, we call them as lumbar vertebrae. How many are there? 5. And number of vertebrae in their hip region, sacral region, lower abdomen, those are, how many are there? 5. And number of vertebrae in your tail region, we call them as coccygeal vertebrae. How many are there? 4. Count them. Count it. 7, 12, 5, 5, 4. Count it. How many got? Number of vertebrae maximum. 33, right? That was in fetus, not in you. In adults, 5 of this sacral vertebrae, 5 of this sacral vertebrae fuse to become only 1. Again, in adults, 4 of this coccygeal tail vertebrae fuse to become only 1. So now count how many bones were there in vertebral column. What is 6? Got it? 26, good, because there was removal of almost, see, 9 minus 7, 2. So 9 bones become 2 bones, 7 got lost, therefore 26. Therefore, vertebrae were 26. Count them. 8 cranial, 14 facial, 1 hyoid, 6 ossicles, 1 sternum, 24 ribs, and 26 vertebrae. How many of there? Total 80. Got it? All these bones were components of your axial skeleton. Guys, understood? Got it? So my question, number of bones in vertebral column in adults, 26. Number of ribs do you have? 12 pairs, 24. Number of sternum, 1. Number of ossicles, 6. Number of hyoid bones, 1. Number of facial bones, 14. Number of cranial bones, 8. Got it? So all together we can get a total count of 80. Those are the bones in your axial skeleton. So 8, 14, 1, 6, 1, 24 and 26. So total was about 80 bones, correct? Done? Very good. Then let us count about appendicular skeleton, appendages. So hands and legs and attached with pectoral and pelvic girdles. So first we'll count number of bones in your hands and then legs, right? So this, guys, when you speak about appendicular skeleton, appendage, what do you mean by the term appendage? Appendage means any kind of hanging structure, possibly a limb. At cellular level, appendages also stand for cilia flagellum, which help in locomotion. So appendages in cockroach or legs, 3 pairs of legs in cockroach, that was also acceptable. Appendages, okay. So appendicular skeleton. They include your forelimbs, hind limbs and girdles. You got anterior girdle and posterior girdle. Anterior girdle, shoulder was called as pectoral girdle, whereas posterior, hip was called as pelvic girdle. Okay, so forelimbs plus pectoral girdle and hind limbs plus pelvic girdle. So all these components, they're almost 126 bones. Let us count it. Your hand, look at your hand. This bone is called as humerus. Right radius, left ulna. See this carefully. Okay, this is the bone, this bone, this bone. This is humerus. And humerus was attached to 2 bones. Radius and ulna. Radius, ulna. Okay, 1, 2, 3. Done. And these radius and ulna are attached to wrist bones. How many wrist bones are there in total? In each hand, 8. There were 8 wrist bones. These wrist bones are called as carpals. How many were there? 8. 8 carpals. And these wrist bones were in turn attached to metacarpals. 1, 2, 3, 4, 5. These were metacarpals. So how many metacarpals were there? 5. And then metacarpals are attached to bones of your fingers. We call them as digits or phalanges. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Phalanges. Done. So count. Humerus 1, radius 1, ulna 1, carpals 8, metacarpals 5, phalanges 14. So what was the total now? 30 bones. Done? Got it? So humerus, radius, ulna, carpals, metacarpals, phalanges. There were 6 types of bones in your forelimbs. 1, 2, 3, 4, 5, 6 types. So humerus, radius, ulna, carpals, metacarpals and phalanges. Got it? 30 bones. Each forelimb has got 30 bones. So 30, 30. 60 was done, right? 2 limbs, 60 bones here. Done. What about your hind limb, legs? We'll discuss that also. In hind limb, there was a largest, strongest, heaviest bone of your body, that was thigh bone, called as femur. It's a ball, femur, and femur was attached to two bones again, tibia, fibula. That's okay. Tibia, fibula. And on top of them, see, between the junction of femur, tibia, fibula, there was a cup-like bone, kneecap, which is found on ventral side. This kneecap is called as patella. Tibia, fibula. And this tibia and fibula were attached to ankle bones. What do you call ankle bones? Tarsals. How many tarsals are there? 7 only in each limb. And tarsals were attached to metatarsals. 1, 2, 3, 4, 5. Metatarsals. And these metatarsals were attached to, again, phalanges. Finally, phalanges. 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. There were 14 phalanges in each hind limb. Now guys, you can count up. So, femur 1, tibia 1, fibula 1, patella 1, tarsals 7, metatarsals 5, and phalanges count to be 14. Count them. 1 plus 1 plus 1 plus 1, 4, 4 plus 7, 11, 11 plus 5, 16, 16 plus 4, 30 bones. Count them properly, Tulasi Lakshmi. 1, 2, 3, 4, 4 plus 7, 11, 11 plus 5, 16, 16 plus 4, 30. You might be having a doubt that hand has 8 carpals, leg has 7 tarsals. Where was the missing bone? Here, right? You do not have patella in hands, you have patella in knees only, therefore 30. So 30, 30 in both limbs, 60. 6, 30, 30, 30, 30. How much was the count now, till now? 120 bones. Okay, so 120 bones is the full count. So fore limbs 30 30, hind limbs 30 30. Now these forelimbs and hind limbs are attached to axial skeleton through special bones called as girdles. Pectoral girdle, this one is made up of 4 bones total. Clavicle, this clavicle, scapula, clavicle, scapula, 4 bones. 2 clavicle, 2 scapula. Whereas pelvic girdle is only made up of 2 bones, hip bones called as coxal bone, coxa. Count them. 30 plus 30 plus 30 plus 30 plus 4 plus 2, 126. Right? There are 120 bones in your limbs and 6 bones in your girdles. 4 bones in pectoral girdle, 2 bones only in pelvic girdle. Therefore, 80 axial, 120 limbs and 6 bones in your girdle. So, total it was almost 6. Guys, you need to first understand how to count bones, then we can get into details of joints, how it was attached to hip bone, how clavicle and scapula form a socket here, how coxa bone forms a socket here. These are details we learn once we know basic things. Do not get into the depths of the subject right now. You still have more time to understand, learn, apply and memorize. Okay? Is it okay till this point? So counted all 126 bones. As I told yesterday, I'll make you count everything. Done. Okay, thank you. That's for today. So signing off, we'll meet tomorrow. Thank you. Happy holidays. Stay home, stay hydrated, be careful. Drink more water. Please be careful. Done.